of death worldwide after the cardiovascular diseases. Among all types of the cancer, lung cancer can cause the more percentages of death. There are several techniques nowadays for treatment and diagnosis of cancer. The first one is surgery, which most of the time is ineffective, especially when the cancer is spreading through the whole body. The second one is radiotherapy, which is done by a very high power ionized X-ray or gamma ray on the body and with damaging the cancer cells. Because of this reason that more ionized rays are used for this technique, it has a very high side effect, and the normal cells can also be damaged by using this technique. The third one is chemotherapy, which is by generating a special chemicals through the body, which can also go through the normal cells, because it's not only for rapid dividing cancer cells this technique can be used, also it can detect all the rapid dividing normal cells. Therefore, this technique has also a very high side effect. The fourth one, which is not very usual to use, is immunotherapy, hormone therapy, and gene therapy. They are more in the research field. In immunotherapy, they try to improve the immune system against the cancer. In hormone therapy, they try to decrease the amounts of the hormones which are connected to some types of the cancer. In gene therapy, they try to remove some damaged genes with the new one that cells can divide and die in the normal way. In the fourth technique, which I'm focusing more on this technique today, is photodynamic therapy. I will go through the more details in the next slides. What photodynamic diagnosis and therapy is? The technique is based on the preferential accumulation of a special drug called photosensitizer, which is very sensitive to a special wavelengths of light in the cancer cell, with subsequent fluorescence emission and production of reactive oxygen species, or ROS. The technique is based on three very important key components. The first one is the photosensitizer, the second one is photoexcitation, the third one is cell oxygenation. Photoexcitation is usually done by special wavelengths of light. It depends on the drug. In which range the drug can be excited, and what is the wavelength that the drug is very sensitive to it. I will explain the photodynamic diagnosis and therapy based on the Jablonski diagram. As you see here, the molecule of the photosensitizer can be excited by the absorption of a special wavelength of light. Then it goes to the first or second excited state. When the molecule wants to come back to the ground state, it can go through an internal conversion without emission of any light, or it can come back to the ground state via the emission of the fluorescence signals. These fluorescence signals helps the researchers to find out where the cancer is exactly generated, because the drug can just be absorbed inside of the cancer cell. Therefore, due to the generation of the fluorescence signals, the researchers can find out where the drug is exactly. It is possible also to that the molecule go to the first triplet state, or T1. During the decay from the triplet state to the ground state, it can go through several interactions, or it can just generate a phosphorescence. It is possible that the molecule go through the interaction with the oxygen and produce some superoxides, which is toxic for the cell, and it can damage the cell, or it can go through the production of the reactive oxygen, which is also very toxic for the cell, and the accumulation of the high amounts of reactive oxygen on the cancer cells can kill the cancer. What I'm going to talk about today is, first, introducing photodynamic diagnosis and therapy by using a novel patented drug called PVP hypericin. There are several drugs with different properties which are used nowadays. But in this talk, I just focus on a special drug called PVP hypericin. The second one is to present the prospect for using PDD and PDT. What is the limitation of this technique? Then I'm going to introduce the aim of the present study, the strategy we used for improving the technique, and experimental setup we used for this improvement, results and discussion, and finally the conclusion of the whole process. Okay, what's PDD and PDT by using photosensitizer PVP hypericin? Nowadays, the researchers try to improve the technique by several things. First, find a drug with a high quantum yield of singlet oxygen generation. Second, invent a drug with high extinction coefficient in the red or infrared region, because if you know, there is a relation between the wavelengths of light and the penetration in the body. We want to improve or increase the penetration of light in the body, because if the cancer is located in a part which is deep inside of the skin, then we need a longer wavelength of light. Therefore, the photosensitizer, which can be excited by using red or infrared light, are better photosensitizers. The third property is they should be low dark phototoxic, that we can just excite it with a special wavelengths of light, and the process of the excitation be under the control. Fourth is the high fluorescence emission. The drug should also generate the high fluorescence signals, that we can find out where the cancer is exactly located. And then it shouldn't be toxic itself by the nature. That makes some side problems during the treatment. With all this, by the invention of a drug called PVP hypericin, all these properties are, they could reach all these properties. PVP hypericin is a complex made of the hypericin, which is the sensitizer, with the PVP or polyvinylamine, and then with the hydrogen bonds between the oxygen groups of PVP and OH groups of the hypericin, this complex is formed. What exactly PVP hypericin does? The first one is it accumulates in the densest region of the membrane. The second property is in the long-term incubation, it can also penetrate into the nucleus. The third property is it can destruct the cell by destruction of the plasma membrane, nucleus, or by doing some damages to the protein and vital enzymes. Here you can see the penetration of the drug in the plasma membrane during the cell division and also during the resting phase. And these are two molecules of the sensitizer in the densest region of the plasma membrane. The prospect for using photodynamic diagnosis and therapy is limited by, first, photosensitizers can also penetrate to the normal cells. Second, continuous excitation results in an expansion of necrosis in cancer cells, which can also have some side effects, because when we have the necrotic cells among the cancer, it can do the rupturing of the plasma membrane, and the content of the cells can go outside of the cell, and it can also make some inflammatory response. The third is immoderate reactive oxygen species generation due to continuous excitation leads to oxidative stress, causing photobleaching and inhibition of photodynamic diagnosis. Because when we have a high amount of photobleaching, then the signals are gone, and we cannot detect the cells precisely in the body. The first is the lack of effective photoexcitation protocol. Nowadays, the technique is doing by using the continuous excitation, as I explained before, which has a lot of high side effects. Therefore, in this project, I tried to optimize the photoexcitation protocol by optimization of the irradiance and also exposure time. What is the aim of the study? The first one is examining the cancer selectivity of the drug, which I explained to you. The second is introducing a method to optimize the excitation via both irradiance and exposure time and cell oxygenation. Third is developing photoexcitation protocol that can replace the continuous excitation in the technique. And fourth is developing a specific excitation protocol that can selectively destroy the cancer with relatively mild side effects on the normal cells. But what is how we do this dosimetry or this optimization? Nowadays, there are several methods for dosimetry. The first one is done by direct dosimetry, which considers the very strong correlation between the generation of reactive oxygen species and photodynamic efficacy. Because they believe that whole technique is based on the generation of reactive oxygen species. They try to monitor the amounts of reactive oxygen, and then based on the amounts of the reactive oxygen, they can optimize the efficacy of the technique. The second method is based on the dosimetry due to the biological response. They try to monitor the cells when they are exciting the drug to see how the cells react to the excitation. And the fourth one is implicit dosimetry, which considers the strong correlation between the photobleaching and also photodynamic efficacy. Photobleaching happens when the drug loses its ability to generate fluorescent signals. It can be due to some damages of the cell, or it can be due to some chemical damages to the drug itself. Therefore, based on the photobleaching and the amounts of photobleaching, they try to optimize the technique. The fourth one is explicit dosimetry. They consider all the parameters which can affect the photodynamic reaction, like light intensity and exposure time. Now the question is, what explicit dosimetry is exactly? Explicit dosimetry refers to the use of all measurable quantities. And it can be done by using this formula, which epsilon shows the extinction coefficient of the photosensitizer, C shows the concentration of the drug in the cells, I shows the light intensity, rho shows the density, H nu shows the energy of a photon. Due to the photobleaching effect, the drug is somehow related to is as a function of the excitation. In many modelings, they consider that photobleaching can be formulated as an exponential decay. If we put this equation in this one, then we can get this formula, which shows that the photodynamic dose is related to the amounts of the light intensity and exposure time. And these two are the very important parameters which should be considered during the treatment. In this equation, when we have the continuous excitation, T is equal with clock time, or the hours which we perform the photodynamic therapy. And in the pulse excitation, it is related to the number of the pulses multiplied with the pulse length. To achieve optimal efficacy of photosensitizer, the applied light intensity and exposure time has to be chosen appropriately. What is the strategy of this study? First one is to use explicit dosimetry to optimize the light intensity and exposure time during the technique. Second is to do some live cell imaging and see how the cells react to the excitation and concentration of the drug inside of the cell. Third is dosimetry due to the biological response. And fourth is to adding a special drug called CFSE, which is non-toxic for the cells, and it just improves the fluorescence signals. The imaging setup we have used is a fluorescence microscope. We changed the optics of the microscope and we put a very low power LED in our microscope for the excitation. The LED can be operated in continuous or pulse mode. And also we used a CO2 chamber during the imaging because the cells we shouldn't have any stress to the cells that they can cause the cell death during the experiment. We should prepare some environment like the body during the experiment. Therefore, we used CO2 sensor. And this is the optics of the beam during the imaging. This shows the beam shape in the focus, which shows that it's latent Gaussian shape, because we should have the same distribution of the light during the imaging. And this is the images of the different pulse lengths and light intensity during the imaging. Now, to find out which light can be used during the experiment, we need to do some spectral analysis. We should find out the spectrum of the drug to find out which light can be absorbed much more than the others. Find out the precise light wavelengths and light range. In this picture, you can find out the absorption spectra with the blue light and the emission spectra with the red light. As you see, there are several peaks. The first peak is around 444, the second is 488, the third is 553, and the fourth is 595. We used two drugs, as I explained. One was non-toxic, the second one was the photosensitizer. Both drugs can be excited with 488 nanometer. Therefore, based on the spectra, we use this light for the excitation. And we have two emission peaks, one in the green region, the other in the red region. This one belongs to the CFSE or non-toxic drug, and the second one belongs to the PVP hypericin or the photosensitizer in the red region. Therefore, the both drugs were excited with 488 nanometer, and based on the different fluorescence signals, we could find out which signal belongs to which drug. Now the results of the experiment. Okay. The first image shows the fluorescence signals were recorded during the experiment under different light intensities and exposure time. The first image shows the the irradiance of 1.59 milliwatt per square centimeter and different pulse lengths compared to continuous. And based on the different exposure time, you can find out the different percentages of photobleaching. Under the continuous excitation, we have a very high photobleaching and also in the very long pulse excitation. But when we have the shorter pulse excitation, the photobleaching is much less. This figure shows the higher light intensity of 6.34, and again the very high amounts of photobleaching under the continuous and long exposure. And the last image shows the highest light intensity of 14.27 milliwatt per square centimeter and the same exposure times. And again, the very high photobleaching under the higher exposure times is recorded. This histogram shows the distribution of the brightness of the images. Now, what we did was to measure the percentages of the cell deaths due to different excitation protocols. As you see from the histograms, you can find out it was first to measure the amounts of cell division and healthy cells to find out the excitation protocol, how can affect the health of the cancer cells. The white bars shows the reference cells and the gray and black bars shows the percentages of the marked cells. As you see, even in the reference cells, which they were marked just with CFSE and it's non-toxic, with increasing the excitation, the number of the cell division is decreasing because excitation is some stress for the cells and it can affect the cell division and mitosis. And with a comparison between the dual marked cells and also the reference cells, we can find out that the drug decreases the amount of division with increasing the excitation protocol. And here you can see the image. This one was taken under the continuous excitation. This one was under the low pulse duration of 1.29 milliseconds. The cells are healthy and they go through the mitosis and finally they divide. But in the continuous case, at the beginning they are healthy. After 60 minutes, they round up, and after 2 hours, they finally go to apoptosis. And now the percentages of apoptosis and necrosis among the cells under the different excitation protocols. By a comparison between the excitation protocols, you can find out that the highest necrotic cells were the highest percentages of necrotic cells were recorded under the continuous and very long exposure times. But when we have the lower exposures at each light intensity, we have the lower amounts of the apoptosis and also necrosis. In this case, which is the irradiance of 14.27 milliwatt per square centimeter, and the pulse length of 131, we have nearly the same amounts of apoptotic cells compared to the continuous case. But the amounts of necrotic cells are much lower, which is a benefit of this excitation. Now to formulate these percentages, we tried to also monitor we try to formulate these amounts of cell death. In this picture, you can find out the percentages of cell death in the y-axis and the total exposure time in the x-axis. You can find out that there is a nonlinear relationship between the amounts of the irradiance and also the amounts of the exposure time and death. This is the formula which fits the data and fits this S-shaped curve. And also we have some thresholds. The threshold dose that the cells can tolerate maximally. These three cases are the threshold, which shows that before these three cases, if we have the light intensity the same as these cases, we don't have any therapeutic efficacy of the drug. And for getting any effective for the dynamic diagnosis, for the dynamic therapy, we should have some light intensity and exposure higher than these amounts. And also the same result as I explained before for the 131 milliseconds and the light intensity of 14.27, which shows that the percentages of cell death is nearly the same as continuous excitation. And maybe this case would be a good candidate for replacing the continuous excitation. We also monitor the amounts of the deaths among the cells in different hours. And in most cases, we find out that 4 hours, performing 4 hours for the dynamic therapy is nearly the same for all cases. Now the question is how we can optimize the side effects, how we can find out these protocols, how they react or how they affect the normal cells. The answer was by mixing the two cell types, one cancer cells and second the normal cells, and do the same excitation protocol and find out how this excitation protocol affects the normal cells. In this image, you can find out the cells, the reference or control cells without any drug. We monitor them for 24 hours to see how they behave when we mix them. And in this part, you can find out the spectra of the cell proliferation, which shows that the mixture of these cells doesn't have any bad effects on the cell types. And now the images of the mixture of cells. If you compare, here are the bright field images, and these two are the fluorescent images. These are the normal human lung cells, and these are the human lung cancer cells. If you compare these two images, you can find out the very high fluorescent signal in cancer cells, but low fluorescent signal in normal cells, which shows that the drug can penetrate more into the cancer cells. Just in the continuous case, because of this reason that we don't have the intervals like on and off the light, then the drug doesn't have enough time to leave the cell, and it is excited more than the others. If you compare the amounts of the fluorescence per cell area in this image, you can find out the higher fluorescence signals in cancer cells compared to the normal cells. Now, we tried also to monitor the mixture of the cells in the longer times. Again, the bright field images and fluorescence images. We also marked the cancer cell, as I discussed before, with additional marker called CFSE that we can separate or we can distinguish the two cell types. And as I explained, this green marker doesn't have any negative effect on the cells, and it doesn't interrupt our study. If you compare the first images, which shows the monitoring of cells using the using the irradiance of 6.2034 and exposure time of 54.5 milliseconds, with the second, which is the light intensity of 14.27 and pulse length of 131 milliseconds, and the last one, which is the continuous excitation, you can find out that after 2 hours, using this pulse length and this irradiance, most of the cancer cells are dead, but the normal cell is still alive. In the second part, you can get the same result, but the more percentages of the deaths among the cancer cells, and the normal cell is still alive. In the last one, which is the continuous excitation, you can find out that both cancer and normal cells are dead after the treatment. Now the percentages of deaths among the cancer and normal cells. With a comparison of these percentages, you can find out that, as I explained before, you can find out that the light intensity of 14.27 and pulse length of 131 milliseconds has nearly the same effect on the cancer cells and much lower side effects compared to the continuous case. Now the curves fitted to the percentages of surviving cells. In the first image, you can find out the percentages of surviving cancer cells. In the second one, you can find out the percentages of surviving normal cells under different light intensity and exposure. With a comparison of these two images, you can find out that after 3 hours imaging, using the light intensity of 14.27 and pulse length of 131 milliseconds, we have about 100%, about 0% cell survival among the cancer cells, which it means that 100% of cells are dead. And after 3 hours, using the same light intensity and exposure, we have about 70% surviving normal cells after the treatment. Okay. Now the discussion. Why this happens? Why the surviving cells? Why the normal cells are still survive, but the cancer cells are dead? And why in the continuous case we have deaths among 100% deaths among both cancer and normal cells? The first one is because of the cellular antioxidant system. The cells have some antioxidant system which, if we have small amounts of reactive oxygen species, they can minimize it and they can survive. And if this antioxidant system doesn't work, then they repair themselves. If the damages are not so high the cells can repair themselves. And now, because we don't have enough time, I will just go through the conclusion. The first, although PVP hypericin has specificity for damaging of cancer cells over normal cells, we found out that light intensity and exposure time has a very crucial role in this technique. The more strength of irradiation is used, the more serious face the normal cells in front of. At all experimental irradiance, cells are healthy and part of them even complete mitosis, and when exposed to pulse lengths of the smaller than 13 milliseconds. This excitation minimizes the photobleaching and provides the long-term photodynamic diagnosis of the cancer cells. Third, continuous excitation leads to high phototoxicity on both cancer and normal cells. Whereas the longer duration of the milder excitation results in less activation of reactive oxygen species in normal cells, which decreases the side effects. And finally, we found out based on our results that 3-hour pulse irradiation using the irradiance of 14.27 milliwatt per square centimeter with the pulse length of 131 milliseconds is an effective alternative to continuous excitation. And this excitation protocol induces about 28% cell death among normal cells and about 99% cell death among the cancer cells. And thank you for your attention.